Hi, my name is Annie Henry, and I'm a graduate student at the University of Denver. Today, I'm going to be presenting some of my dissertation research to you. This project is entitled Cover of an Invasive Tree Co-Varies with Environmental Factors to Explain the Functional Composition of Riparian Plant Communities. In general, my research deals with this big picture question: what happens to plant communities when Tamarix is defoliated by the biocontrol beetle Diorhabda? So far, studies have shown a wide range of plant community responses to defoliation, including no change in understory plant community, small increases in native vegetation and diversity, shifts towards more of an upland community rather than the typical riparian species, and increased secondary invasions. So what causes such a wide range of responses? In other words, what are the mechanisms underlying plant community response to Tamarix cover? To answer this question, we're looking at plant community response using traits rather than species identity. Looking at traits makes sense given that a plant's traits, in large part, are what determine its ability to thrive in a given environment. So traits are the direct link between abiotic conditions and plant community structure. Environmental change, such as flow regulation, invasive species establishment, or invasive species removal, changes the environmental filters operating in a community. So it changes the environment. And this in turn changes the traits that are advantageous in that plant community, finally resulting in a community made up of different species. So looking at traits rather than species sheds light on what abiotic conditions are really driving the community. Why does this matter? Well, this is helpful for anticipating plant community response to invasive species establishment and removal, including anticipating the traits of secondary invaders, and then selecting appropriate species for revegetation. Ultimately, the plant community contributes to ecosystem functions such as habitat provisioning and nutrient cycling, so it's really important that we understand what's going on. This study looks at a snapshot of Tamarix cover across a large geographic area and asks what traits are most important in defining riparian plant guilds. And by guilds, I mean groups of species with similar traits. Secondly, how does guild cover vary along a gradient of Tamarix cover and abiotic condition? And lastly, how does guild diversity respond to Tamarix cover and abiotic condition? So we selected 95 sites ranging from low Tamarix cover, more native dominated plant communities, to plant communities with overstories made up entirely of Tamarix. Sites were along the Green River, Colorado River, and the Dolores River, and tributaries connected to those. And all of these sites included in this study had exposure to biocontrol beetle between 5 to 9 years. So these were all part of a larger study published in 2017, looking at the response of vegetation to Tamarix removal across the southwestern United States. For our purposes, we examined all sites in that larger study that only had biological control removal of Tamarix. And vegetation data was compiled by Gonzalez and others and was sampled between 2012 and 2014 using line intercept method. For this study, we compiled data on 17 traits for 139 species present in our sites using online databases and literature searches. This includes true biological traits that primarily relate to resource acquisition and reproduction, as well as what might more accurately be called ecological affinities, so ordinal values that give an overall measure of the plant's response to things like drought, flood, etc. So we chose these traits based on previous studies that have shown them to be relevant in defining riparian plant communities. We also collected data on abiotic factors, elevation, precipitation, river width, distance to river water's edge, soil electroconductivity, and elevation above the water channel to try and parse out the influence of Tamarix cover and these factors. So question one, what traits are most important in defining riparian plant guilds? So the first step in this process is just identifying and defining the plant guilds that are present across the study area. To do this, we used a cluster analysis that grouped species with similar traits. And to visualize them, we plotted the guilds as well as the weighted averages of all the traits in a principal coordinate analysis, or PCOA. And a PCOA is simply a way of ordering multivariate data into 2 dimensions so you can see relationships among different variables. So what you see here is the first 2 axes of the PCOA. Each point is a species and the symbol is its guild identity, and we'll get into the guilds more in a minute. The text represents the weighted averages or centroids of trait values. So the farther out on an axis a trait is, the stronger it is in defining that axis. And I only plotted the top traits here, so there are a whole bunch of traits that were also just clustered around the center that aren't important in defining the plant community. The blue text represents variables that were not included in the analysis, but we calculated the centroids after the fact to help interpret the results. The main result here is that reproductive traits, such as the ability to reproduce asexually, represented here by clonal, seed weight, and perennation, so perennial versus annual, are major factors in defining plant guilds. As are resource acquisition traits like life form, height, and specific leaf area. That's what this stands for. Our cluster analysis identified 9 guilds defined by differences in those traits. And here I'm just going to talk about the 7 understory guilds. A primary distinction in these guilds was the ability to reproduce asexually. So we have non-clonal guilds that can't reproduce asexually and clonal guilds that can. These are radial plots that just show a summary of average trait values for each guild. So the further out on this little spiderweb line is, or point is, the higher that trait value is for that guild. So the non-clonal guilds all tended to have moderate to long bloom periods and light seeds. They were further divided by annuals and perennials. The clonal annuals were further divided by graminoids and forbs. So we have annual graminoids, which were anaerobic tolerant, for example, barnyard grass, and then annual forbs that were drought tolerant, for example, common kochia. Now on the perennial side of things, these 2 guilds were differentiated primarily by specific leaf area. So we had a guild of high specific leaf area species. These are the resource acquisitive species. For example, narrow leaf plantain. And then a resource conservative guild with a much lower specific leaf area. And broom snakeweed is an example species in this guild. There were 3 different clonal guilds. First, we have drought tolerant perennials, such as Canada thistle, anaerobic tolerant perennials, for example, common reed, and lastly, a resource conservative guild with super low SLA, for example, greasewood, so more upland species. So question 1, what traits are most important in defining riparian plant guilds? First, reproductive traits, which tend to be strongly linked to disturbance, such as flooding and fire. This suggests a gradient of disturbances present throughout the study area. Second, height-specific leaf area and life form were important, suggesting that there's also a wide range of drought versus waterlogging tolerance, as well as resource acquisitive versus conservative strategies that are successful throughout the study area. So my second question, how does guild cover vary along a gradient of Tamarix cover in abiotic condition? To answer this question, we used a redundancy analysis, or RDA. This is essentially like a regression analysis, but with multiple explanatory and response variables. So the position of an explanatory variable along this axis is what shows what defines this axis. And then the position of the guilds shows what sites have a higher cover of those guilds. Together, this shows the relationship between guild cover and the environment. In this figure, I'm starting out with just the environmental factors that define these axes. So you can ignore these little red numbers. And the length of an arrow is relative to the importance of that variable. So at high levels of RDA1 here, we have sites that are far from water, have high Tamarix cover, both absolute and relative, and high elevation above water level. So these sites are far away from the elevation and precipitation, so that means they have lower elevation above sea level and lower precipitation. So these are our dry Tamarix dominated sites. Okay, so I should mention here that elevation above sea level is also negatively correlated with temperature. So over here, the low sites are also hotter. Now on the other side of the RDA, this RDA1, we have the opposite types of sites. So high elevation above sea level, higher precipitation, closer to water, and low Tamarix cover. Now I'll show you where the guilds are on this figure. Okay, so the guild points on the RDA are showing you where the sites with high cover of each of these guilds are positioned on these axes. So here we're looking at non-clonal guilds, so only sexually reproducing, and you can see that 3 out of 4 of these guilds are associated with sites far from water and higher Tamarix cover. So these 3 over here, these 3. We were able to do a follow-up analysis with mixed effects models, and we were able to show that annual forbs increased significantly with Tamarix cover. So there's some stats for you for that. So this guild actually has the highest number of invasive understory species also. These tend to have an annual life cycle, reproduce with many small seeds, and have a moderate to long bloom period. Additionally, these species tend to have a higher specific leaf area than one would expect in a dry, resource-poor environment. So this suggests shade tolerance. So these are the types of traits we find in more Tamarix-dominated sites. On the other side of things, we have the clonal anaerobic tolerant perennials, which were, we were able to do a follow-up analysis with that and found that they were significantly correlated with both Tamarix cover and distance to water. So these anaerobic tolerant perennials are more common in wetter sites with lower Tamarix cover. And then the clonal drought tolerant perennials were related to distance to water here, but not strongly related to Tamarix cover. So this suggests that as Tamarix is removed and sites become hotter and drier, this guild may increase, which incidentally is also made up of a fair number of invasive species. They just tend to be different kinds. They are longer lived, more drought tolerant, and can reproduce asexually, excuse me. So for the whole RDA that I just showed you, we explained 31.5% of the variability in guild cover. And this is just to show you that almost 9% is just explained by Tamarix alone. So while Tamarix does covary with environmental gradients, it also has an influence on the plant community separate from the environment. So how does guild cover vary along these gradients? First, Tamarix cover favors a short life cycle, so annuals with lots of seed production and shade tolerance. Drought-tolerant perennials are likely to replace annuals as Tamarix is removed. And the typical Riparian guild clonal anaerobic tolerant species are negatively affected by Tamarix. So while Tamarix does covary with environmental variables, it creates a filter independent of other environmental factors. So last question real quick, how does guild diversity respond to Tamarix cover in abiotic condition? So this figure shows that axis of RDA1 from before, but plotted against guild diversity, measured as Shannon diversity's index for guild cover. So what we found was that mid-ranges of both Tamarix cover and environmental conditions are associated with higher guild diversity. So in here we have the highest guild diversity. So very dry as well as very wet sites have a much lower diversity, and the same is true of Tamarix cover. Very high Tamarix cover and very low Tamarix cover have lower diversity than intermediate levels. So this relationship was confirmed with a quadratic mixed effects model, and the RDA1 explains 43% of the variation in guild diversity. So how does guild diversity respond to Tamarix and abiotic condition? Basically, plant communities are more specialized at both low and high levels of Tamarix, as well as environmental extremes. Okay, so the take-home messages of this study are first, that we've highlighted this role of Tamarix as both a passenger and a driver of plant community function. Tamarix facilitates non-native species because of their drought tolerance and shade tolerance and their high reproductive output. As Tamarix is defoliated, understory plant communities may shift more towards those clonal drought-tolerant guilds. And lastly, native species from that guild could be chosen for revegetation in order to compete with non-native secondary invaders. So it looks like I probably have a few minutes for questions. Just want to acknowledge the funding sources and people who have helped out on this study, and I'll take some questions. Thank you.